Hello, my name is João Sequeira, and today I'll be presenting my review on Marcos Louro's master's thesis, A Stochastic Model of Centriole Assembly. So, I'll start by talking about the motivation behind the thesis, followed by the problem and the objectives to be achieved. I will then show the methodology developed and show the results obtained. I will then show the conclusions reached and show some of the proposals for the future work to be developed. The regulation of the number of centrosomes is crucial for the cell's division process, as anomalies in it can lead to serious diseases, such as cancer, as a result of them dividing way too much. This regulation is mainly related to its main components, the centrioles, and its ability to only duplicate once per cell cycle. The protein that is thought to be the main regulator of this duplication is the PLK4 enzyme. However, and despite the mechanisms of this protein being well known, the relationship between the number of centrioles and the PLK4 concentration is yet to be discovered. So, to ensure the cell central duplication process faithfulness, it was proposed that there is a structure, the cartwheel, made of Cas6 dimers, which has a one-to-one relationship with the number of centrioles. These dimers could result in an annular structure, similar to the one shown in the figure, and can pile up on top of themselves to form an annular pile structure whose activity is related with the PLK4-1. Nevertheless, the most important thing to take away from this is that if the piling up is predominant in relation to the production of new structures, the cell will be able to ensure that it only duplicates itself once. But is the piling mechanism efficient in controlling the number of cartwheels and hence the number of centrioles? This is what this master thesis is intended to find out. Okay, and how was this achieved? So, two stochastic models were defined to describe the cartwheel's creation. One, which considers the cartwheel's creation as the end product of a series of linear steps, and another, which takes into consideration the more complex dynamics of oligomerization and piling up of the Cas6 proteins. Nevertheless, it is crucial to mention that the validity of both methods relies on two key assumptions. Firstly, the models assume that the cartwheel formation is the determining step in the control of the number of centrioles. And secondly, it is assumed that the cartwheels have a composition which is based on a 9-fold rings of Cas6 which can pile up indefinitely. Okay, so moving on to the results. The analysis of the first model showed that it was necessary to be a relatively high stacking rate for the stacking of the Cas6 molecules to prevent the formation of new cartwheels. The analysis of the second model showed similar results regarding the stacking rate, and moreover, it allowed to see that when the production of Cas6 is increased, the system doesn't respond linearly, creating less new structures than it would otherwise, as can be seen in the figures, where despite the high influx or production of Cas6 molecules, the number of cartwheels, as represented in the black line, doesn't increase linearly. Okay, so the second model also predicted that the cartwheel's elongation, contrary to the creation of new ones, is linearly related with the increase of Cas6 molecules, as can be seen in the figure. This means that a slight increase in the concentration of Cas6, or PLK4, will probably lead to a pile elongation instead of an increase in the cartwheel's number. Furthermore, it was observed that in concentrations propitious to the creation of supranumerary cartwheels, an asynchronous synthesis is represented in the difference in the average length of the individual structures. This basically means that the sooner the cartwheel is created, the more time it will have to elongate. So, moving on to the conclusions. The results obtained from the model show that theoretically, the proposed mechanism can explain how cells control the number of centrioles according to the extrapolation that these correspond to the number of cartwheels. Furthermore, the model analysis also suggests that the cartwheels have a length distribution, something that isn't usually mentioned in the articles and that might be relevant physiologically. To test experimentally the predictions obtained by the models, it is suggested the manipulation of the PLK4 Cas6 expression. This way, the protein's influence in the length and the number of cartwheels can be evaluated. However, measuring the length of a cartwheel is something that might actually be difficult to do. Hence, it is suggested a method which uses the distribution of the aspect ratios of random sections of the structure, as the average aspect ratio can be used as an estimator, at least within a given range of values. This was the bibliography used, and finally, thank you for the attention.